diversity is more in the Varanasi region, and many new different vegetable crops are also been cultivated. So in that context, they have shifted to Varanasi. Then it has been promoted to Indian Institute level during 1999, and this is the view of our institute. I mean, my past institute. Okay. Then in that institute, they are working on several vegetable crops. This is the mandate vegetable crops, major vegetable crops. That is 20 crops they are working on as a major vegetables. That is belongs to solanaceous, leguminous, cruciferous, cucurbitaceous, malvaceous, and root vegetables. And in addition to that, they are recently they started working on the minor vegetable crops also. Almost 22 minor vegetables, it includes leafy vegetables, then some leguminous minor vegetables also like winged bean, lima bean, faba bean, and other goats also. That is ivy goat, round melon, then spiny goat, summer squash, snake goat, and baby corn also. So these are the different crops we are working on, and our institute is rich in diversity of vegetables. You can see how much diversity we are having over there, and not only in these four crops, on all vegetables we are maintaining almost 6,000 germ blossoms. They are maintaining over there, and they are having rich source of genetic material they are having. So any materials they can access for a breeding purpose or for screening purpose, we can access over there. So with this background, I will start my actual lecture. That is, my lecture is on emerging viral disease of vegetable crops in Indian scenario. So let me have some introduction. These viral diseases are becoming a major threat for vegetable cultivation. As we all know about this viral disease, if we go for any vegetable crop, we can see that some one or another viral disease will be associated in the crop. So in India, at present, more than 300 viruses are reported to be infecting the vegetables, and this list is increasing every day because of the evolutionary mechanism of the viruses. These viruses are evolving by three different processes, either by mutation or recombination between the different strains of the same virus or between the different virus, and reassortment of the genome, that is exchange of the genetic material between the two different viruses. And also the as the viruses are transmitted through polyphagous vector, this polyphagous insect vector they are visiting so many crops. As the name says, the polyphagous, it will be infecting the so many crops. So from one crop to another, it is easily transmitting by these insect vectors. And because of this, the host genes are also becoming expanding every day. And and another one way is introduction of genetic materials. We are introducing the new crops from the other countries. So through that also the viruses are being introduced, and through the seed-borne nature of this virus, they from the foreign countries it is being introduced here. So this is the reason why the viral diseases are becoming major threat for the vegetable cultivation. And as far as the vegetables are concerned, till now we are depending on the private companies only for the resources. But now this scenario is changing, as the introduction of the genetic materials are restricted from the foreign countries. So now we can restrict somewhat the viral introduction. And now we will see the what all the different viruses are emerging in the vegetable crops. As this begoma virus, this is a major virus among all the viral genera, and it is the largest plant virus, as it is occupying more than 445 species in this begoma virus genera. Earlier, Partivirus was the largest genera. It was having 200 viral species, but now it exceeds 445 species and become the largest plant virus. And it is having the nature of twin and icosahedral particles, that is encapsulated inside the twin particles and transmitted by the whiteflies in a circulative and non-propagative manner. That means viruses will acquired by the insect vector at one time, and it will take some time for uh, circulate inside the insect body, and then only it will transmit. So these are all the this transmission nature we need to know, because it will help us to decide the management practices. And the major diseases associated with this begoma viruses are leaf curl diseases in many crops, including solanaceous and okra also, and yellow vein mosaic disease in okra, then yellow mosaic disease in leguminous vegetables, then yellow and so on. This begoma virus is infecting several vegetable crops. Almost we can say all crops, and then this disease incidence caused by this begoma virus is ranged between 90 to 100% during the dry season. During this dry season, the plant will have less water content, and the sap tastes much sweeter than the wet season. So insects uh, plants during the dry season, so their perpetuation also will be faster. So the during the dry season, the disease incidence will be more than the wet season, and loss is estimated up to more than 60% loss because of this virus. And now we will see the different tomato viruses which are reported from the India, which is causing associated with the leaf curl disease. It has been first reported from 1995, and then till 2021 within India itself 14 different tomato leaf curl viruses were reported, and across the globe there are more than 100 tomato leaf curl viruses are there. And within India we can see the New Delhi virus, Karnataka Bangalore virus, Gujarat virus, Palampur virus, then Pune virus, Rajasthan virus, Patna virus, Kerala virus, then Gandhinagar virus, Jodhpur virus, then Mumbai virus. Likewise 14 different viruses were reported on the tomato leaf curl disease alone. So you can see that symptoms are also so much extremities. We can see this is highly infected plant, and this is somewhat initial stage of the infection. So entire morphology of the plant itself will collapse. And along with this virus, there are associated with the beta satellites also, and these aggravate the symptoms of the main virus. And sometimes they are all associated with another DNA B particle also. If it is associated with the B particle, then they are bipartite, and if it is only A alone is there, then they are monopartite. So few viruses are only bipartite, and almost all are monopartite viruses. And in the North Indian scenario, these are the major challenges we are facing over there in the growing the tomato crop. See, these are in this slide we can see both biotic and abiotic stress. So during the June to September, this is the main season. We start the planting, and June to September we are facing the we used to get the more of rainfall also, and during the time temperature also will be more, so we used to get hot and humid condition which gives the plant wet heat. And during the December we will have the extreme December and January we will have the cold stress. Temperature will go up to 8 degrees Celsius, 7 8 degrees Celsius, and night temperature also will be I mean night temperature only 8 degrees Celsius, and day temperature will not exceed 20 also. So and during March to May we will have the dry to 47 degrees Celsius. So during this time, we can see different biotic changes, biotic stresses. We can see during this July to October, we can see the leaf curl, and during the March to April also. So almost here 4 months and here 3 months, almost 7 months we can get the leaf curl over there, and we can say it is a hotspot for the leaf curl disease. And before this cold, November to December and January to February, we can get the virus over there. So this is the viral scenario, major viruses in the North Indian condition as far as concerned. And in addition to that, other late blight we can see, and fruit rot by alternaria, and crown rot by alternaria only, and then bacterial spot we can observe over there, and early blight also we can see. See, this early blight and this fruit rot, crown rot, all are caused by the alternaria only. But during the October and November, we can get the early blight symptom, and during this time, that is February to April, we will get the fruit rot and collar rot symptoms. So this is for information I am saying. It is not related to virus, but we can see the viral diseases, that is leaf curl disease, is more during the July. Almost 7 months we are getting the viral leaf curl disease over there. And similarly on chili, it has been first documented in 2007 only, that too from India only. And till 2015, we are having almost 7 different viruses were associated with the leaf curl disease on chili crop. And after that, only very few reports were there. Then tomato leaf curl Jodhpur virus on chili also, and both in inland and island condition. Chili leaf curl Gonda virus, that is also reported during 2017. In the same way as that of tomato, here also they are associated with the mostly with beta satellites also. and only two viruses are bipartite, all others are monopartite only in case of chili leaf curl disease is concerned. We can see the extreme, this is a healthy plant and infected plant. Because of this leaf curl, we used to lose the yield like anything over there. And every year we won't get there. Alternate year or 2 years once only we are getting the symptom over there. And the virus associated with these symptoms are also changing every year. Initially it was chili leaf curl viruses were there. Later now it become tomato leaf curl. Jodhpur virus is predominant than the chili leaf curl virus. So it is changing every year. And then begoma virus on cooker bits, you can see several, this is a very few list I have given. In addition to this, several other viruses are also associated with the cooker bits which are causing mosaic disease. Earlier these symptoms were thought of associated with the cucumber mosaic virus. Then later we diagnosed that it is because of the begoma virus only. Like on all cooker bits we are having the begoma virus infections. And on similarly on okra, the two different symptoms were there. One is yellow vein disease, another one is okra initiation leaf curl. So very commonly we used to see in South India is the yellow vein disease only, but in northern India is concerned we used to have the okra initiation leaf curl. So in this symptoms you would have not seen here. You can see the typical symptoms. Cupping of leaf will be there. If we see the hopper infection also, we will have the cupping symptom. But here in case of cupping, you can see on the lower side of the vein won't be uniform. It will be somewhat undulated or rough nature we can say. And we can see the enations, small leaf-like outgrowth on the lower surface of the leaf. That too, if we keep the leaf towards the sun, and if you see, we can see these different colors. Can you able to see here? You can see the small leaf-like outgrowths, and the twisting of petiole also will be there. Petiole won't be straight, it will be twisting. So in case of normal plant, the twisting won't be there. So, and if it is infected with the inhalation leaf curl disease, the plant won't produce fruit, or even if it produce fruit, it won't have any seeds, and it affects the yield drastically. And it affects at all stages of the crop. But if it is infecting at the later stage of the crop, then yield loss won't be that much. But early stage, it is causing complete yield loss. And in case of yellow vein, it won't stop the fruit production, but it will produce the unmarketable fruits. So different viruses were been identified associated with these two viruses. I mean two symptoms in India, like bendy yellow vein mosaic virus, Asian leaf curl virus, like yellow vein Maharashtra virus, New Delhi virus. Likewise, several viruses are there. And in other crops like legumes and radish, then basela also we are consuming. Here we are not consuming as a leafy vegetable, but in North India it is one of the major leafy vegetable. And in carrot, amaranth, these are all the different begoma viruses were documented on the vegetable crops, other vegetable crops. Is cucumber mosaic virus. This cucumber mosaic virus earlier scenario was very different. Now it is gaining more importance. This is infecting several crops and it is prevalent throughout the world. That is in all the conditions, the temperate, tropic, and subtropic regions. And it is transmitted by the aphids in a persistent manner. I mean non-persistent manner. And it is icosahedral particle, that is somewhat circular particle of 29 to 30 nanometer size will be there, and it is causing yield loss of 10 to 20%. And it is having not only through this aphids, it is being transmitted through mechanical and also seed. So due to the seed transmission nature, it is having more importance now. And it is a RNA virus. Only begoma virus is a DNA virus, and all other virus what I am going to say are I mean RNA viruses. So earlier this was restricted only with the cooker bits. That very less incidence was there, less than 5% or 10% like that. Then it has shifted its host to other vegetables like tomato, brinjal, chili. And in nowadays this in tomato is concerned, this CMV is becoming so serious problem. I will tell you at the end of this presentation. Then these are the different symptoms caused by the CMV on different vegetable crops. You can see the mosaic symptoms on cucumber, and you can see the mosaic on snake gourd also, and you can see the healthy and infected fruit. It is changing the fruit shape also. This shape the fruit, and it is causing chlorosis also in bottle gourd. and in case of chili, it is causing mosaic. And in case of tomato, it is causing the filiformity, that is shoestring like symptoms will be there. Sometimes this used to confuse with the herbicide injury also. 2,4-D spraying also will cause the same kind of symptom, but we have to identify it carefully. And almost on 16 different vegetable crops in India, it has been documented, CME has been documented, and the reports are here. Next is orthotaspovirus. This is another one important virus. And earlier, the orthotaspovirus was more prevalent in the southern India rather than northern Indian conditions. And nowadays, it is there also it is emerging as a problem. And this is transmitted by the thrips vector, and it is also RNA virus, which is having tripartite genome. That is three fragments. Genome of the virus will be fragmented in three different fragments, and it is also somewhat circular in shape and of 120 nanometer, 80 to 120 nanometer size, bigger than the CMV. And in case of the thrips transmission, they will acquire the virus at the first and second instar larvae. And then when it acquires it the second and first instar larvae, then only they can able to transmit it in the adult stage. They can't even transmit at the, if it acquire at first stage, they can't transmit in second stage. Likewise, so it needs to build up the virus tighter inside the vector, then only they can able to go for transmission. So this helps us in devising the management. So if we see the first and second instar larvae of the thrips, if we go for taking up the management strategies, so we can able to control this very we can it is possible to control, and it is causing the yield loss of 29 to 100% yield loss also been recorded. In India, 7 different taspovirus species has been documented on different vegetable crops, and that is groundnut bud necrosis virus, watermelon bud necrosis virus, capsicum chlorosis virus, tomato spotted wilt virus, iris yellow spot virus, peanut yellow stunt virus, and melon yellow spot virus. This peanut yellow stunt virus alone is been documented only on groundnut. All remaining all other viruses, 6 viruses were documented in vegetable crops in India itself. Then we can see the different symptoms caused by the groundnut bud necrosis virus on tomato. See, we can see this is a typical symptom of the taspovirus infection. We can see the concentric rings, that is rings inside the ring we can see, and with the green center. The same symptom will be confused with the alternaria also. Alternaria also will have the concentric rings, but they won't have the green center here. As we see here, green center, there will be completely brown. And the same way you can see on fruits, we can see the circular concentric rings on the fruit surface also. And on stem, we can see the necrosis. And but here we won't see the concentric rings. We won't see. But in case of alternaria, we will see the concentric rings like that shooting board symptom will be there. And on leaves also we can see necrotic lesions with a marginal browning will be there. Later it leads to drying of the plant. Now, in southern India is concerned, this is having more than 50% incidence we can see in the field. But in case of northern India, it is almost maximum we can see 20%. That too, every year we are not getting this disease. Alternate year or 2 years once only we can see this. And in case of brinjal and chili, the same groundnut bud necrosis virus and capsicum chlorosis virus are causing the chlorosis disease. We can see the concentric rings on leaves of brinjal, and also necrotic spots also we can see on the brinjal leaves. We can see here clear symptoms. And here we can see beautiful symptoms of capsicum chlorosis virus infection on chili, that is concentric rings. Then circular ring spots with the green center on chili leaves. We can see the symptoms here. And this is necrotic lesions on the stem. Next, watermelon bud necrosis virus. This was first documented in India during early 1990s. That too from southern India, that is Karnataka. And later, now it is spreaded to entire country. Now in northern India, we could not able to take a crop. Even single crop, it is very difficult to take because of this virus alone. So most of the farmers, they stopped growing the watermelon crop in northern India. And most of the watermelon we are getting from southern part of the country only. So cost of fruits also high because of transportation, everything. So different symptoms associated with this disease are, we can see bud necrosis, that is necrosis from the growing tips, and it will extend downwards. Later it leads to drying of leaves. and marginal necrosis of leaves, and necrotic spots on leaves. And sometimes even we won't see the necrosis, even chlorosis also we can see with this watermelon bud necrosis virus. Can see the paleness of leaves, and on fruits also we can see the chlorotic spots, and see the healthy and infected fruit size, and the lesions also we can see. Not only on mature fruit, in immature fruit also we can see the symptoms like necrosis, necrotic lesions. Then this is a recently emerging problem in tomato. In northern India, last year we observed this symptom. Initially, the symptoms were like chlorotic spots were there. Then the chlorotic spots increases, and then necrotic lesions were also there with circular spots with green center. And on back side, if you see the marginal, I mean venal necrosis with necrotic spots were also there. And later, plants started wilting. And on stem, if we see closely, necrotic lesions were there. And on petiole, and petiole also we can see the necrosis. Then necrosis on the growing region. And after by coalescence of these lesions, it become drying of leaves. Then you can see the purplishness of growing buds, and again the bronzing of leaves. On fruits also we can see the symptoms. So we initially we were thinking that it is because of groundnut bud necrosis virus, and but we tested, it was negative for groundnut bud necrosis virus, then capsicum chlorosis virus, and also even TSWV, that is tomato spotted wilt virus. And it was positive for the watermelon bud necrosis virus. So previously in that, this was a symptom collected in the polyhouse. In that polyhouse, previously the bitter gourd crop has been raised. So next they have raised the tomato crop. From that it has been shifted, and the strain was very virulent strain, and it is killing the entire plant. And even incidence was even 100%. They have replanted the entire polyhouse planting, and then only they got some incidence. I mean some yield they got. And this is also emerging as a new problem now. And only very few reports earlier it was made in Maharashtra. One report was there, and Karnataka some sequence they have deposited, but as a complete disease report now we have documented from IAVR, this one. And on cowpea, in case of southern India, we used to consume cowpea as a legume crops, that is pulses only. There they will take as a vegetable crop. Entire pod they will immature vegetable, and immature pod they will consume as a vegetable. And in that also this groundnut bud necrosis virus is a major problem over there. Almost we will get a 20 to 25% incidence we will get over there. And the symptoms you can see here: chlorotic spots with venal necrosis, and you can see the chlorotic spots with green center, and chlorotic lesions. This is TSWV emergence on tomato. This is in southern India. We are not seeing in the northern India this disease, and the symptoms are exactly similar to the groundnut bud necrosis disease. But we can see the difference only in the fruit infection. In case of groundnut bud necrosis, we will see the chlorotic lesions, but in case of TSWV on tomato, we will see the necrotic lesions here, and the fruits are completely unmarketable, and it will become very hard. And we can see that severe infection on the fruits here. And this kind of necrotic lesions we won't observe with the groundnut bud necrosis. This is associated with only TSWV. This is also emerging in southern India, that too documented in Karnataka. In some part of Tamil Nadu also it is there. And these are the publications showing the importance of emergence of this TOSPO virus in vegetable crops. Now, next one is the party virus, and this is also the largest group next to begomovirus. This is the largest group of plant virus, having more than 195 species, and it is monopartite and ssRNA virus. And unlike CMV and orthotospo virus, it is a flexuous rod shape. They are all circular icosahedral particles. They are flexuous rod shape, almost around 800 nanometer. That is very bigger than the those two viruses. And it is having a character inside in electron microscopy. If you see the cell, we can see the characteristic pinwheel inclusion bodies. These are the aggregation of this virus particle in a certain fashion. So this is a typical nature of this party virus, and then it is transmitted by aphids in a non-persistent manner. So it has to acquire the that to silent bond. It has to acquire the virus, and immediately within 1 or 2 minutes it has to transmit. So this is so barrier cropping like management strategy we can use for this party virus, because yeah, it has to acquire the virus at virus and immediately it will transmit. So if we have the barrier crop in between the infected and main crop, so the virus will be acquired by the aphids, will sit on the barrier crop and they will leave the virus there itself, and then when it moves to the healthy plant, it will be free from virus. So these are the things we need to know to devise the management strategies. And different party viruses associated with this vegetable crops in India are bean common mosaic virus in several legumes, papaya ring spot virus in cucurbits, then pepper mottle virus, chili venal mottle virus, watermelon mosaic virus, zucchini yellow mosaic virus, zucchini tigre mosaic virus. And these two viruses on chili and other viruses are all infecting the cucurbits. And not only through aphids, they are all transmitted through mechanical transmission also. And very few viruses like zucchini yellow mosaic virus, they are seed borne. They are not having any vectors, they are seed borne also, and incidence ranges from 20 to 95% on different vegetable crops. Can see the different symptoms caused by the party viruses. That is PRSV. Whenever this party virus is there, we can see the blistering symptoms on the leaves. This kind of blistering symptoms are there, then we can assume that they are infected with the party virus. So these are the different symptoms on pumpkin, rich gourd, cucumber, and bottle gourd. Here you can see the as we see in the papaya fruit ring spot, we can see the ring spots on the leaves also. And this is here you can see the blistering on snake gourd, vein banding symptom. You can see the banding around the veins. This is also caused by the PRSV on snake gourd. And this is zucchini yellow mosaic symptom, yellow mosaic virus symptom on snake gourd. You can see the yellow spots are there. And on fruits also, it is having effect. This is the healthy fruit and infected fruit. You can see the malformation of the fruit and size of the fruit are also reduced drastically. It leads to unmarketable these fruits. Next, zucchini yellow mosaic virus on pumpkin. You can see the mottling, mosaic mottling, and yellow mosaic on bitter gourd. And in this symptom, you can see circular spots are also there, yellowing of leaves also there. This is having a mixed infection of both zucchini yellow mosaic and papaya ring spot virus. And this is a zucchini tigre mosaic virus infection on gherkins. You can see the mosaic pattern, but based on the PCR only we have detected. By symptom wise, it is very difficult to differentiate. And this is bean common mosaic virus on cowpea. And then this is mottling symptoms on the cowpea. This is caused by cowpea aphid-borne mosaic virus. These two symptoms. And this is chili venal mottle virus. Here also you can see the mottling around the veins. So this is typical symptom for chili venal mottle virus. This virus is not common everywhere. It needs a somewhat low temperature. Either in northeastern region or Bangalore region, we are getting this symptom. On other places, it is very difficult to observe this problem. So it is problem only in the colder climates, hilly areas, in capsicum or polyhouse grown chilies. It is a problem. Next one is the Tobamo virus. So this Tobamo virus is the oldest virus, and it is a highly stable virus in the environment. And it is a rigid rod shape. Like party virus, it is flexuous rod shape. This is a rigid rod shape, and it is of the size only 300 nanometer. There it is 800. Here it is only 300 nanometer. And these are not having any vectors. They are mainly transmitted through mechanical transmission only, either through the implements or other cloth materials or trellis wires. That is contaminated materials only. They are mostly they spread through, and they can even survive on the leaf debris also for longer time, and even on soil surface also for longer time, since they are very highly stable. And yield loss is around 10 to 15%. And the similar along with the CMV, this is also seed borne in nature. And because of the seed borne nature, these two viruses, that is CMV and Tobamo virus, are gaining importance nowadays in these vegetable crops. Different vegetable infecting Tobamo viruses are tobacco mosaic virus, tomato mosaic virus, cucumber green mottle mosaic virus, then pepper mild mottle mosaic virus are prevalent in India. These are the different symptoms. The difference between the mosaic and mottling symptom. So I need to explain here. You can see the in case of mottling, there won't be any distinct shapes won't be there, or there won't be any start or end. In this symptom, you can see this is mottling. So where this the light green, dark green started, where it is ending, we can't have some definite shapes or definite pattern won't be there. So this is called as mottling symptom. In bottle gourd, this is caused by CGMMV. In pumpkin, then cucumber also. In tomato, this is causing mosaic by tomato mosaic virus. And the same on fruit also, you can see the discoloration of the fruits. And not only this fruit discoloration, the fruit become very hard and they won't have any juice at all. It is not fit for marketing. And these are the different reports showing the emergence of this tobacco virus in the vegetable crop is concerned. Next, it is a recently emerging problem, that is poliovirus and cucurbits, and it is a problem throughout the globe and transmitted by aphids. And this is also RNA virus. And this first poliovirus documented on cucurbits is cucurbit aphid-borne yellow virus, which is having the yield loss of 40 to 50% in France. That too, this is during 1980s. It was documented first time. These are the different symptoms, yellowing symptoms. You can see on bitter gourd, yellowing of leaves, and on watermelon also you can see the yellowing, and on squash, then pumpkin. The typical symptom for this poliovirus is not only yellowing, you can see the downward rolling of the leaves will be there, and leaf will become leathery, and marginal rolling of leaves downwards will be, that is a very typical symptom for this poliovirus. As the name says, polio, potato, leaf roll. So same way, the rolling of leaves will be there under downwards, and leaf will become thick and leathery, and brittleness will be there. So this is a typical symptom for this poliovirus. And first in India, it has been documented during 2017 from Karnataka, followed by in Tamil Nadu also by this department only. And later in 2021, it has expanded its host range to different crops. You can see squash, ivy gourd, sponge gourd, pumpkin, satputia, ridge gourd, and watermelon. In 2023, still some more crops: muskmelon, round melon, snake gourd, bottle gourd, and ash gourd. In this list continues, and so more than 14, 15 vegetable crops are being cultivated in India, and almost all crops are getting infected by this virus. And in this poliovirus, not only cucurbit aphid-borne yellow virus is prevalent in India. In addition to that, Peppo aphid-borne yellow virus and melon aphid-borne yellow virus. These two are also there. They are very less. That too only in UP we have documented. In other parts of the country they have documented only cucurbit aphid-borne yellow virus. And next one is the Crini virus. This is also recently emerging, and first documented in India by during 2021. After that, in 22 we have documented on bitter gourd, cucumber, and watermelon. And in 23 again we have documented on some more crops also. And this Crini virus is also RNA virus transmitted by whiteflies in semi-persistent manner, and yield loss is up to 63%. And not only this Crini virus also causing the yellowing disease. Compared to here, if you see in this case, downward rolling of leaves will be there, but in this case of Crini virus, veins will remain green, and intervenal chlorosis will be there. It is exactly confusing with the deficiency symptom. This sulfur deficiency, iron deficiency will have the intravenous chlorosis. Same symptom will be there, but in case of deficiency, we can observe on the younger leaves. But in this Crini virus infected one, they will be symptom will be observed on matured leaves. We can't see any symptom on the younger leaves. Only on matured leaves we can see the symptoms. And you can see the yellow vein symptoms on here also. Veins are remain green, and intravenous chlorosis we can see here also. Veins remain green here. And different Crini virus species infecting cucurbits are cucurbit yellow stunting disorder virus, cucurbit chlorotic yellows virus, beet pseudo yellow virus, and lettuce infectious yellows virus. In this, only these two viruses are being documented in India. That is CYSDV and CCYV. And you can see the different crops we have surveyed, and different types of yellowing symptoms. Here, these are all crinivirus infected, and here this is poliovirus. And these two symptoms are not, we suspected for crinivirus, but they are not for crinivirus. They are positive for some other new virus. That yet too we have to confirm it. And you can see the poliovirus and crinivirus combined infection, downward rolling of leaves also there, and intervenal chlorosis also there. So this is very major problem in across several vegetable crops. We can see here almost 10 different vegetable crops I have listed here, like ash gourd, bottle gourd, cucumber, ivy gourd, then muskmelon and pumpkin, then satputia, round melon, then ridge gourd, snake gourd, and squash. This is the document which is showing the emergence of this coronavirus and poliovirus. In addition to this virus, this tobacco streak virus also been documented on nearly 14 different crops in India, but they are not that much very here and there. The reports were there. They are not having any that much economical importance. Very few reports were there, and one or two reports, that's all. Not at the major incidents were not documented. Only reports were made. These symptoms are almost confusing with the taspovirus symptom, that is orthotaspovirus symptom. And on okra, chili, onion, and legumes, this virus has been documented. And these are some of the lists of the tobacco streak virus infections. And other virus in northeastern region, they have documented the Maclura virus. This is also one of the partyvirus genera, Partyviridae genera, that is large cardamom chipkey virus on chili. And the symptoms are like mosaic mottling only we can see here. They were suspected for leaf curl, but they ended up with the identifying this new virus. During 2018, they have documented on northeastern region. So these are the some of the emerging problems of viral diseases in India. And recently in 2020, during the COVID time, yeah, a new virus problem was outbroken in the Maharashtra region, and it created nearly about 60% of the crop has been lost within 10 days. So they have named it as a Tiranga disease. So the fruits have become, they lost its complete natural color. They will become uh, like this. They will become, that is color. Uniform color won't be there, and like bleaching of that red color has been observed, like yellow, green, and red color. And this has been named it as a Tiranga, that is three color, during in the Ahmedabad, Pune, and Nasik region. And it was they were suspecting for some other cause, but it was finally they identified it as associated with different viruses. That is cucumber mosaic virus, then tobacco vein distortion virus, then tomato mosaic virus, groundnut bud necrosis virus, and tomato chlorosis virus. These are the 5 viruses are associated with these different samples. So in this CMV is the main cause for this hardiness of the fruit. You can see the complete malformation of the fruit here, and there is no juice at all. Complete flesh only is there. There is no juiciness is there. That much hard, and the farmers have faced so much problem over there. From IIHR, they have identified this is associated with 5 different viruses. So same way in Punjab and Himachal region, in tomato crop has been found to be infected with this associated with these 9 different viruses: tomato leaf curl nudilly virus, CMV, GBNV, different potato viruses, then tomato chlorosis virus, and tomato mosaic virus. You can see the different diversity of symptoms on the tomato crop they have seen during their survey. And in this tomato chlorosis virus they have documented for first time in the country. Earlier outside the country it is there. This is also a crinivirus only. It is documented on tomato crop. During last year only they have reported this one. Not in this, New Delhi virus is having more than 65% incidence, followed by the potato virus, that is party virus. It is around 25%. And mixed infection is also very common. The 2 virus, 3 virus, 4 and 5 virus, up to 5 virus infections were commonly they have diagnosed with this viral disease of tomato. And in conclusion, as far as the virology is concerned, vegetable is a, we can say, a boon. There is no limitation for virus study. So many viruses are there. Even if you take any crop, any that too in cooker bits itself, it's endless. We can, that many new viruses are emerging as a problem. So many scopes is there. Scope is there for viruses, and we need to have only we are doing up to the diagnostics, but still we have to go in depth regarding the etiology, how it is being spread and what is the interaction with the viruses and the vector. So much scope is there. And it's still it is not at fully it is still in nascent stage only. We need to have good work in this one. Thank you. And I acknowledge in this moment my chairman. So from